Hi, my name is Ronnie, and this past summer in the Phillips lab, I investigated how plasma catecholamines, specifically norepinephrine, may help explain the effects of epidural electrical stimulation in individuals with spinal cord injury or SCI. Now, imagine trying to sit up or stand and suddenly feeling dizzy because your blood pressure can't stabilize. Well, that's a daily reality for many living with SCI. Now, when someone moves from lying down to sitting or standing, blood redistributes to the lower body, meaning less blood returns to the heart, so blood pressure drops. Sensors called baroreceptors located within the heart, as well as the carotid sinuses in the neck, will detect this change and quickly alert the sympathetic nervous system, which is the body's fight or flight response. In turn, the adrenal glands will release norepinephrine into the bloodstream, which is a chemical that helps to stabilize blood pressure. This will then increase heart rate as well as cardiac output and begin to tighten the blood vessels, restoring blood pressure within seconds. However, in those with high-level SCI relating to injury in the neck or cervical region, those communication pathways from the brain to the remainder of the body are disrupted. So without enough norepinephrine release, the body cannot correct the drop in blood pressure, leading to orthostatic hypotension, which is when there is a sudden fall in blood pressure that causes dizziness, fainting, and long-term cardiovascular risks. To help restore this lost control, our lab studies a therapy called electrical epidural stimulation, or EES. It involves surgically placing a small electrode in the epidural space, which is the area just above the spinal cord, that delivers tiny electrical pulses to reactivate the circuits controlling blood pressure. Now, we typically target the T10 to T12 region, also known as the hemodynamic hotspot, because it contains the highest concentration of neurons that regulate blood vessel constriction. In essence, EES helps the spinal cord remember how to control blood pressure when the brain cannot send the signal. Now, previous work from our lab has shown that EES can successfully defend against severe blood pressure drops when an individual moves upright. Now, while we know that EES improves blood pressure stability as well as orthostatic tolerance, its underlying mechanisms are poorly understood, so identifying this link will help to clarify the mechanisms by which EES restores cardiovascular control in SCI. This led to my main research question: does EES increase levels of norepinephrine in the blood during orthostatic challenges in those with high-level SCI? To test this, we recruited 3 participants with high-level SCI as well as known blood pressure instability. Each individual was then implanted with an EES device targeting the T10 to T12 region of the spinal cord. Now, we used the head-up tilt test, which is a safe and controlled way to mimic standing up. Now, the table slowly tilts upright to the desired angle, usually around 60 degrees, which causes blood pressure to drop. Now, we conducted this under 2 conditions: when stimulation was off and then when stimulation was on. This allowed us to directly see how EES changes both the body's chemistry as well as its physiological changes through blood pressure responses. For each test, we collected blood at 3 time points: before the tilt while lying flat, 1 minute into the tilt, and 30 minutes post-tilt after returning to rest. These samples were used to measure circulating norepinephrine levels in the plasma, allowing us to track sympathetic activity. During this time, we also continuously monitored blood pressure to evaluate real-time physiological changes and to see not only that EES worked, but how it worked. Now, without EES, norepinephrine levels remained quite low throughout all phases of the testing period, rising only slightly from 0.2 to 0.38 nanomoles per liter, with a total difference of 0.18 nanomoles per liter, ultimately depicting a weak sympathetic response. With EES activated, norepinephrine levels were significantly enhanced throughout the entire testing period, rising from 0.25 to 0.75 nanomoles per liter, corresponding to an overall increase of 0.5 nanomoles per liter. Looking at the blood pressure trace, we can see how these changes play out in real time. With stimulation off, blood pressure drops sharply and remains low, depicting orthostatic hypotension. When stimulation is on, blood pressure rises and remains stable well into the tilt and recovery period. Now, this mirrors the increase in norepinephrine, showing that EES strengthens sympathetic activity and helps regulate blood pressure more effectively. The key finding was that EES boosts norepinephrine levels and keeps them elevated even after stimulation is off and orthostatic stress has passed, meaning that EES may not only correct blood pressure in the moment, but also promote longer-term recovery of autonomic control. In summary, EES increases sympathetic activity by enhancing norepinephrine release, stabilizing blood pressure during orthostatic stress. Now, this therapy may serve as a first-line approach for cardiovascular dysfunction in spinal cord injury, enabling greater independence without dizziness or fainting. Finally, I would like to thank the members of the Phillips Lab for their support and guidance, as well as CanTap Talent for funding this project.